Good morning, everyone. Today we will have a second seminar from our English seminar series, and our guest is Jennifer Yuzon, who will be talking to us about her projects and work in fungal biology. Jennifer has a PhD in plant pathology from University of California, Davis, and works as a postdoctoral researcher and data scientist at Berkeley Lab. She's also a bioinformatics expert with extensive knowledge of space and radiation biology, microbiology, plant pathology, supervised machine learning, and diverse coding languages. Jennifer has 8 years of computational biology experience working in government and academic settings with a team of scientists and engineers of diverse backgrounds. Jennifer is eager to continue answering important research questions and working on complex dataset and systems. Let's welcome Jennifer and listen to her talk.
Okay, yeah, thank you everyone for tuning in today for my seminar, and special thanks to Adriana for the invite. I really appreciate the opportunity to talk about my research on fungal radiation and resistance. So Adriana went over my background, but just to reiterate, I really enjoy fungal biology, especially in the context of the environment, and that's how I landed in the Naval Research Lab working under Zhang Wang. We did some collaborations with NASA, looking at the fungal response to space conditions. And for my work at the Naval Research Lab, I studied a lot genes and gene networks and how they evolve under irradiation stress, both on Earth and in space. So at the center, you'll see an image of a gene network, and it's kind of messy, but from here on out, I'll try to distill the information and make it more digestible for the rest of the presentation. So while at the Naval Research Lab, working with Zhang, we focused on properties of fungi that protect against irradiation. So there are a lot of fungi that are isolated from high radiation environments, such as Chernobyl and the International Space Station. And many fungi that are isolated from these sites produce melanin, which is a pigment well cited in the literature as being protective against high radiation. Also, research from our lab identified DNA pathways as strongly protective against irradiation, and we focused a lot on homologous recombination and non-homologous end joining. So the reason why fungal radiation resistance is such an active field and really an interest in the Wang Lab is that we're interested in the application of these fungal phenotypes. So we're hoping to apply what we know about fungi to protecting space and military personnel during space exploration and in conflict zones. Also, we're interested in the production of food, material, and medicine during space travel and establishing life in space. So for my talk, I will be discussing important genes and their pathways involved in radiation resistance. So first, I'll talk about 2 projects in which we study fungi in the lab, and we look at specific proteins, the melanin pigment, and DNA repair pathways. However, we later tried to apply our findings to our space programs. So I was lucky enough to have led 2 NASA collaborations, one involving the Artemis mission and another related to Mars-like environments. So the first project, I'll talk about this directed evolution experiment of fungi under increasing doses of gamma radiation. So in the lab, we study a fungus called Exophiala dermatitidis. It is highly resistant to radiation. One of the properties we are interested in is melanin as a protective mechanism against radiation damage. So this project is actually a continuation of my colleagues' work, Jillian Romsdahl and Zach Schultzhaus. They evolved several strains of wild-type isolates, which are melanized, and then several strains of a PKS mutant, which are unmelanized. And they use this directed evolution approach, which cycles between gene diversification or mutagenesis and then screening for useful gene variants that we're interested in. Later, they would look at the genome for mutations and try to see if they were actually adaptive for radiation resistance. So the figure at the bottom, this shows the different isolates that they identify that are highly adapted to radiation after the directed evolution experiment. And the isolates are listed on the x-axis, and on the y-axis was the survival measurement compared to the wild-type control. So the wild-type control didn't experience any radiation. So yeah, they identified several isolates, and a lot of them were in the unmelanized, the PKS population. So though these evolved isolates grow very well under high radiation stress, when they're grown in normal conditions and they're co-cultured with their controls, they don't grow as well as the unevolved isolates. So there seems to be a trade-off between adaptation to high radiation stress and also normal functions. In these isolates, Jillian and Zach, they identified mutations that shut down the non-homologous end joining DNA repair pathway. And this pathway is highly error-prone in repairing DNA. I should mention that the most significant DNA damage that is caused by ionizing radiation are double-stranded DNA breaks. And they found that by shutting down this more error-prone non-homologous end joining pathway, they increase the survival of their fungal cells. This is because, again, likely because non-homologous end joining pathways are inaccurate in DNA repair, leading to cell lethality. So Jillian concluded that more accurate DNA repair pathways, homologous recombination takes over, and there's a high fidelity DNA repair, and this increases cell survival. So to continue their work, I focused on a larger panel of isolates and looked at both gene expression and genome integrity. So it's a multi-omic analysis. There were 13 samples that were identified to be resistant and resistant to high radiation in the evolved population. And we took 3 replicates to measure irradiation and non-irradiated condition for each of these isolates. This resulted in 78 transcriptome profiles. And then for the genomics project, we used again, from the directed evolution study, there were 9 evolved and unevolved isolates that we focused on. So the unevolved isolates were the control, and we exposed them to acute radiation, so 8,000 gray, and took 9 random colonies to check the genome integrity after this acute radiation dose. And then this generated 81 genomes. So this was a rather large collaboration with the Joint Genome Institute, and it was a very big data analysis project. So here we're looking at a Go enrichment analyses for genes that were highly expressed during irradiation. So on the x-axis are the irradiated, are the evolved populations and the unevolved populations. And this is, again, a comparison between their irradiation response versus their control response. On the y-axis are Go ontology terms, which are genes that are enriched in the expression profile. Wild type C and PKSC are the original isolates, so they have not been evolved under irradiation. And the wild type 15 are, again, the wild type melanized isolates that have evolved under high radiation stress. And the PKS evolved, or the PKS 15 population have been evolved under radiation stress and do not produce melanin. So we could see that in our results that consistently all the isolates upregulate DNA integrity genes such as replication, binding, and repair. The downregulated genes didn't really show any consistent pattern, but this general binding term shows up. So this indicates that DNA expression is really important for irradiation, for the irradiation response. So we know that all the isolates, whether they're evolved or control, that they express DNA repair. But next, I want to see what are the evolutionary changes or adaptations that are going on in the evolved population compared to their control. So therefore, I compare the gene expression between the evolved versus the unevolved isolates. First, I started looking at differences under non-irradiated conditions. So we can see in the wild type evolved populations versus the controls that they highly upregulate DNA integrity genes. And in the PKS evolved population versus the PKS control, they upregulate more mitochondrial and redox balance type genes. So why would they upregulate these genes under non-irradiated conditions? We think that this expression pattern indicates that adaptation led to a more constitutive response in the evolved population, and they're using this non-irradiated condition as a recovery phase. Then I want to look at the PKS evolved versus the wild type evolved population. So the unmelanized versus the melanized evolved populations to see if they had any different strategies. So the comparison shows that the PKS evolved population favors this redox balance or oxidoreductase genes, whereas the wild type evolved population are enriched for DNA integrity genes. So this indicates that the PKS evolved and the wild type evolved population have different strategies for irradiation recovery. So next, I looked at the evolved population versus their control under the irradiation conditions. So they were exposed to 1,000 gray. Unfortunately, I didn't see any consistent patterns or strong patterns for upregulated genes and also for downregulated genes again. But I did see that there was a binding term and that differentiates the PKS evolved versus the wild type evolved population on the last row of plot A. So I looked back at the gene count analyses. So I'm not showing the data here, but it seems that there are only a few genes that are differentially expressed in the evolved population versus the control under irradiated conditions. Instead, the greater number of differentially expressed genes are under non-irradiated conditions. So this led my group to hypothesize that the evolved isolates, their adaptation response is to constitutively express genes for repairing the cell and the DNA, even when not irradiated. So the adaptation occurs during non-irradiated conditions. So a lot of DNA-related genes were upregulated during irradiation in many of the isolates. So what I did next was look at the genome and try to identify if there was different patterns in genome integrity between the evolved and unevolved populations and their controls. So on the x-axis, you could see that we have the different wild-type control, PKS control, wild-type evolved, and PKS evolved population. And on the y-axis are the number of mutations after 8,000 gray exposure. And each point is an average of 9 replicates per strain. And each panel represents a mutation type. So on the left are structural variants, and these are large mutations that cover hundreds to thousands of base pairs, and they come in different types, such as deletions, duplications, inversions, and translocations. And on the right, we have point mutations, so only a single change in the genome, single base pair change, and this is called a single nucleotide polymorphism, a SNP. The control populations are in blue, and the evolved populations are in red. So I did a Kruskal-Wallis test and performed this for each mutation type, followed by a done test with p-value correction. And the significant p-values are shown by a star. So the SNPs didn't show any major differences between population. However, for structural variants, we see that the PKS control has many more mutations. The DNA integrity isn't as strong as the other isolates. And this indicates that melanin does play a role in protecting the DNA. However, the PKS evolved population, they have much lower mutations. So likely from directed evolution, they evolved compensatory strategies in the absence of melanin. So the transcriptome profile indicated redox balance, antioxidants to protect the cell and the DNA. So what are the causes of higher fitness in these evolved populations? I tried to identify next some candidate genes for molecular analyses. In this table, it shows a list of genes that had mutations in either the coding region or around regulatory elements. And these are the mutations that only occurred in the evolved population, and they occurred multiple times. So this is parallel mutations at the, parallel mutations. These mutations occur multiple times independently in the evolved isolates, indicating adaptation potentially to high radiation stress. So I found that Jillian and I found that there was one gene that we call 1778 that had multiple mutations, and this 1778 gene encoded a transcription factor. In the PKS evolved population, it's downregulated, and in the wild-type population, it's upregulated. So the mutations in the wild-type population are actually highly disruptive, such as stop gain and frame shift, indicating that this transcription factor is non-functional, even though it's highly upregulated. So coming back to how I think about genes and gene pathways, I did a gene network analysis, and I looked at the top 5 highest correlated genes with 1778 in terms of their expression profiles. And the top most correlated gene has a function associated with chromosome transmission during replication and segregation of the sister chromatids. But also other genes are related to the replisome, indicating that, again, DNA replication, binding, and potentially repair are very important for the radiation response. So my colleague Jillian, she further examined the effects of this transcription factor on radiation resistance. She had a wild-type strain, deleted 1778, and exposed the mutant to higher doses of radiation. This mutant was slightly more resistant to gamma radiation under a wild-type background. However, the most mutations that occurred at 1778 are found in the PKS evolved population. So in the future, we'd like to examine this gene under a PKS unmelanized genetic background. The takeaways that we got from the study was that the evolved strains of Exophiala dermatitidis developed different expression profiles compared to the control strains. So wild-type evolved population emphasized DNA repair, binding, and replication, whereas the PKS evolved population emphasized oxidative stress response. And more genes are differentially expressed during non-irradiated conditions in the evolved isolates. So this indicates that the cells are actively repairing themselves and also the DNA during this recovery phase, the non-irradiated phase. So, and then from the structural variant or the mutation analyses, we saw that the PKS control population, it had higher number of large mutations than other population, indicating that melanin does protect DNA. So the second project that I worked on involves another type of protein called 8115 that we studied in the lab, again in Exophiala dermatitidis. So yeah, we have a lot of studies using Exophiala dermatitidis and studying them under different radiation conditions, so UV and gamma radiation. And we see that during the expression, the transcriptome analyses, there was an 8115 gene that was highly expressed. And in the lab, these 8115 mutants, they're highly susceptible to gamma radiation and UV radiation. So on the right, we have a phenotypic assay. There's the wild type, the 8115 mutant, and then a mutant with 8115 reconstituted back into the genome. So under control conditions, there's no difference in growth, but with higher gamma and UV radiation, the 8115 disruption mutant doesn't grow very well. Next, we want to see if there were any unusual properties related to this 8115 protein. So we looked at whether the protein was able to fold or was intrinsically disordered. So here at the bottom, there's a graph showing this type of analyses. On the x-axis are the amino acid positions, and on the y-axis measures the level of disorder or order. So anything above 0.5 is being disordered and values below 0.5 are ordered. And we see that throughout most of the protein that it's highly disordered, it doesn't fold. So it's kind of unusual for these proteins to not fold. And in some of the literature, these proteins are evolved in stress and protection against desiccation, and this is especially so in the tardigrade literature. So my colleague Zach, he did some preliminary microscopy studies, and he showed that the 8115 N-terminus part of the protein, when it's tagged with GFP, it's likely expressed in this. It's expressed in this nuclear circular organelle that we think is the nucleus. He tried the full fusion, so the full protein with GFP, but he had difficulty expressing the protein, so he used the N-terminus. And then what I did to look further into the nuclear localization properties of 815, I used the NucPred software to identify if there were nuclear localization signals. So here is the protein sequence. It's more than 900 amino acids long, beginning with the N-terminus and ending with the C-terminus. Warm colors indicate strong homology with other NLS or nuclear localization signals, and cool colors indicate that there aren't any strong matches. So we see that there are 3 NLS motifs near the N-terminus and 1 potential NLS in the C-terminus. And these have high homology with other eukaryote nuclear localization signals, including mammalian systems, which I'll come back to later. So a deletion mutant of 815 was generated, and we tried to do some differential gene expression analyses. However, we didn't come up with very strong patterns, indicating that gene expression is without 815 or with 815 is more nuanced in the cell. So what I did next was to do a gene network analysis. So what are the genes that are co-expressed with 815 to try to gain some information about the biological relevance of this protein. So I used different transcriptome profiles of Exophiala dermatitidis from different projects with genetic backgrounds of the wild type, the PKS, the 815 mutant, either under irradiated or non-irradiated conditions. And this type of analysis, it does pretty well in describing gene networks with at least 19 transcriptomes. Here I used 90. And 21 gene modules were described. These gene modules are groups of genes that are inferred to work together and have similar functions. But I won't go into too much detail about each one. I'll focus more on the gene module in which 8115 plays a significant role. So 815 was found in a gene module that was highly enriched for terms related to DNA replication, binding, and repair. So again, this DNA integrity aspect is very important. And this module is highly correlated with irradiation. So here is a table of the different genes that are correlated with 8115, and these are the top 5 genes. It also shows the correlation with irradiation, so the correlation is quite high. And the annotations show that, again, these genes are related to DNA replication, DNA repair, DNA binding. So we have 2 primases, 2 polymerases, and again, a chromosome transmission fidelity protein for the proper segregation of sister chromatids during cell division. So based on these data, we think that 815 does interact with DNA, not just from the gene network analysis, but also the nuclear localization signals and our microscopy studies. And I just want to quickly mention that 8115 didn't have any annotations. We didn't see any Hox domains. So likely this protein uh, binds DNA, but not in a specific manner. So there are a lot of bioinformatic and phenotypic results for 815 related to DNA repair and nuclear localization. Therefore, we'd like to examine if 815 directly binds to DNA, and also if it protects mammalian cells and yeast cells, and if it localizes to the nucleus of mammalian cells. So to answer these questions, we'd like to express 815 in E. coli, purify the protein, and then incubate it with a plasmid, and then use a gel mobility shift assay to see if 815 binds DNA. Next, the group is working to transform mammalian cells and then expose these mutants to X-ray radiation and measure survival. And also, to, again, try tagging 815 with GFP and to see if the GFP signal and 815 localizes to the mammalian nucleus. So it's an ongoing project, and now I'd like to talk more about our space program. So there were a lot of findings about fungal survival in the lab, and now we want to see if our results can be applied to our space programs. So I was lucky enough to collaborate with NASA and lead this fungal project for the first Artemis program. So the first Artemis launch was uncrewed, and the rocket orbited around the moon for more than 20 days. And the rocket's trajectory took our fungal samples above lower Earth orbit, and it launched in November of last year. And then for Christmas, I got the samples back and started processing them right after the mission. So the purpose of our study was to identify if DNA repair and melanin protects fungi from radiation in deep space, and the Artemis project was a great opportunity to allow us to identify if these genetic and molecular properties of the fungal cell allow them to survive and adapt to lunar conditions. So I'm just going to go over our experimental design. So at the beginning, I put fungal spores into a dissolvable capsule and placed them at the bottom of a 50 ml Falcon tube and overlaid this with 40 mils of media, so that by the end of the mission, when I got back my samples, I had mycelial growth of the fungus throughout the medium and then spores at the very top. So the spores, I was able to harvest them and also do some phenotypic analyses, so looking at how they grow in culture, and then subculture the spores for transcriptomic and genomic analyses. And then for the mycelia, that I used that for biomass to study if there were differences in biomass between the different strains, and then use the proteins for proteomic analyses. The media, I extracted metabolites and submitted them for metabolomic analyses. And I'd like to mention that there were 20 samples, so 5 of the wild type, 5 of a non-homologous end joining mutant, and 5 of the homologous recombination mutant, 5 of the unmelanized mutant. So again, coming back to our interest in DNA repair pathways and melanin protection and how they apply to irradiation protection. So we're still processing the ground samples, but we're really excited to continue with this project and hopefully get some interesting results. Next, I'd like to talk about another NASA collaboration that I led. So we sent our fungal samples to the Antarctic because 39 kilometers above the Antarctic space is this sort of Mars analog environment, and we want to test how fungi survive under these conditions. So for this project, we were again interested in the high radiation stress above the Antarctic, which is similar to the Mars radiation profile. So we are sending our fungal samples to the Antarctic on a balloon, which will fly above the continent's surface. And our samples will be in a container called a Mars box, and it's equipped to monitor UV and ionizing radiation in addition to temperature. And the ultimate goal is to identify qualities in the fungus that can be used to protect humans in Mars environments, and also to produce biomaterials during deep space exploration. So the table on the right, you could see that the UV radiation profile is similar between Mars and the 39 kilometers above the Antarctic. And it's different from Earth's surface. And ionizing radiation is also high under these conditions. So again, the Antarctic space, 39 kilometers above the Antarctic space, highly replicates this Mars analog environment. And for this project, we are testing 4 types of strains, but this time in Aspergillus niger, which is a different fungus, and it's highly amenable to these space experiments. We have our wild type, and then also our non-homologous end joining mutant, homologous end joining mutant, and then also our melanin deficient mutant. So coming up, so we're repeating our interest in DNA repair and melanin production. At the center, you could see this is a diagram of the Mars box, and located under the shutter, there is something called a T-Rex, and it's the actual holder that contains the samples. So the T-Rex can hold up to 64 quartz discs, and on the discs will be fungal spores that will be dormant during the space flight, but which we will revive after the mission. So half of the samples will be shaded and receive only ionizing radiation, and the other half will be exposed to both UV radiation and ionizing radiation. And we'll repeat the experiment in the lab as well as on the ground. And this will allow us to tease apart the effects of UV versus ionizing radiation, and also space versus Earth conditions. So this project hasn't happened yet. Hopefully it will happen at the end of this year or next year. But we're really excited for this project. So I went over a lot of different projects, both on the ground and in space. And I have to say, this involves a lot of different people at the Naval Research Lab and at NASA. And I want to acknowledge their awesome contributions, especially my mentor, Zhang Wang, and my colleagues, Jillian and Zach. They have been on multiple space projects and continue to support the NRL space program. And with that, I'm happy to take any questions.
Jennifer, thank you for a very interesting presentation and impressive amount of projects that you've managed to work throughout your career. Congratulations on that. And with that, we're going to ask you a few questions. First of all, could you please tell us more about how the irradiation experiments are done in practice? What facility, and how do you prepare your samples?
Yeah, so we do have facilities at NRL where we could expose our samples to high radiation stress. We have a cobalt pool, and we also have a machine that does other types of radiation like X-ray. And, you know, NRL, it has a lot of resources, so we could just easily set up our experiments and our samples in the lab. Yeah, and we use different types of fungal growth. Sometimes we use spores that are dormant. Other times we use cells that are active, so actively dividing. Yeah, I hope that answers the question.
Yes, thank you. You also mentioned that after the irradiation, DNA integrity genes were upregulated. Did you discover some novel DNA integrity genes?
Um, so 815 was a novel discovery that my colleague Zach zeroed in on. And it was, a really interesting protein, difficult to work with in the lab. And then also some of the things that I discovered in my gene network analyses that may not be novel, but that we didn't expect were genes related to DNA replication, like the mini chromosome maintenance protein. Usually that's for the replosome, and we didn't expect it to be differentially expressed or involved in this irradiation response.
Perfect, thank you. And how do you expect these genes to help? And you also did mention bioproduction, so radiation resistant strains for bioproduction. Do you think those proteins could somehow help human radiation resistance?
Yeah, so it's kind of looking far into the future, but we'd like to maybe get some of the melanin pigment and apply them to humans, or maybe I don't know, it's probably far out there to apply 815 somehow as just like a lotion or something for protecting astronauts. That's kind of far out in the future. And in terms of bioproduction, so fungi, they're used for a lot of therapeutics and food on Earth. And if we can get these fungal cells to survive high radiation stress, especially on the Martian equator, we could get them to also not just survive, but also to produce biomaterial. So that's kind of the idea, the far out idea.
I personally am also a huge fan of biotechnology and space, so I'm with you on these far-reaching ideas.
Yeah.
And we have a slightly different question here. So since the project that you were describing to us were conducted with U.S. government institutions or their U.S. government projects, do you accept international students studying outside of the U.S. to do master's degree thesis in these projects? If yes, how the students could apply for such projects and positions?
Oh man, that's a difficult question. It really depends on the immigration policies by each administration, so it changes. And I don't want to, give a definitive answer because who knows what will happen in the future. But generally, if you have a green card, you could study at NRL, but it's always easier if you're a US citizen, again, because times change. I think it might be easier to apply for a master's or some graduate program when for a university, I think. And universities are great. And Adriana, I think you know more about this.
Yes, it's a bit tricky. Yes, so getting in the institutions here in the US, it's a bit tricky, but it's not impossible to get permissions and clearance to work on very specific projects. So like Jennifer said, it's rather institution dependent. So I would advise looking at the websites of the institutes that students or you are interested in, and send emails with questions to get clarifications. That's probably the best I can answer that question.
Okay, yeah.
Do we have any questions? Oh, yes. Another question is, are scientists exposed to any radiation during their experiments in lab? If yes, how do you protect yourself?
Oh, hopefully not. At least from our protocols. So our cobalt pool is in this deep water pool, and so this kind of the water kind of absorbs the radiation, and then for the different machines in the lab, they're kind of hefty structures. So basically, I just open a door and then I put my samples in and then close it, and then it receives some sort of X-ray radiation or UV radiation. Yeah, so probably not. And there's personnel at NRL that test the level of radiation and make sure that everything's up to code.
Yes, hopefully you guys are keeping everyone safe there. So now we have an opinion question. So in 2007, in Plus One, there was a publication about turnable fungi that actually are feeding on the radiation, not just using the melanin as protective dye. What is your opinion about that paper?
Well, yes, I've heard of this paper. I don't think I have a very strong opinion, but it's interesting. Radiation is a type of energy. Potentially other organisms can, use different sources of energy. Yeah, I don't really have an opinion. It's interesting.
Okay.
Yeah.
Another question. So in the first experiment, someone is asking if they understood correctly that you were looking for something that will protect DNA better than melanin.
Or something that complements melanin. Yeah, DNA integrity is a very strong response, and I think it's the first response that evolved in the tree of life to protect against high radiation doses. So I think they all complement each other. I guess I would say that DNA integrity is very important. But it's always great to have melanin and to have other properties in the cell to back it up. Yeah.
Perfect. Thank you for answering the questions. Are there any last minute questions for Jennifer before we thank you for her time and the presentation? Okay. It seems like we don't have any more questions. So once again, Jennifer, thank you for your time and thank you for joining us today and sharing your experience and work.
Okay. Thank you so much, Adriana. Thank you for this opportunity, and have a great day.
Thank you. Bye, everyone.